In the final lecture of the biochemistry module, we're going to go into bioenergetics and the regulation of metabolism. So we'll start off by talking about thermodynamics and bioenergetics proper. Most biological systems are considered open. One thing you do have to take note of, though, is that biological systems are not in equilibrium. If a biological system ever does reach equilibrium, the organism will die. There is no work done on a closed biological system, and physiological concentrations are generally less than the standard concentrations you'll see in the lab. Free energy calculations must also be adjusted for pH. The ATP has a lot of different roles in the cell. Primarily, it's the main energy source. It's a mid-level energy source, and it provides energy through hydrolysis and coupling to unfavorable reactions. The phosphates that are also removed can act as donors as well. There's ATP, adenosine triphosphate, ADP, diphosphate, and AMP, monophosphate as well. There are biological oxidation and reduction reactions as well. They can be represented as half reactions, much like you saw in general chemistry. Most of these reactions have an electron carrier of some sort, for example, FAD or sometimes NAD. The majority of this lecture is going to go into a discussion of metabolic states, so let's begin there. The first is the absorptive state, or postprandial. This is when insulin secretion is the highest and glucagon and catecholamine secretion is decreased. Anabolic metabolism is the most common. This is often called the fed state because this is after we've eaten, so we've got a lot of food going around, meaning that we've got a lot of glucose floating around. This is why insulin levels are going to increase so that we can start to absorb the glucose that's floating around in our system. The fasting state, or the post-absorptive state, is shortly following the fed state. Insulin secretion decreases, and glucagon and catecholamine secretion will increase as the excess glucose is going to want to be stored away. This is observed during some short-term fasting, most commonly during sleep, and there's a transition to catabolic metabolism that occurs, meaning that instead of breaking down nutrients, we're breaking down the stuff within our body, for example, glycogen, as an energy source. The last one is starvation, or prolonged fasting state. You're going to see dramatic increases in glucagon and catecholamine secretions as we try to break down our internal structures for energy sources. Most tissues will convert to using fatty acids for energy or ketone bodies if it's absolutely desperate. Take note that the brain uses ketone bodies for energy but cannot use fat. Now that we've talked about the three states, we're going to go into some hormonal regulations of metabolism. The main relationship is the antagonistic relationship between insulin and glucagon and how they refer to blood glucose levels. This is a very, very popular topic on the MCAT, so really make sure you have this antagonistic relationship understood. Insulin is going to decrease blood glucose levels by increasing cellular uptake. It'll also increase anabolic metabolism rate, meaning that it's going to increase the rate at which external nutrients that are absorbed in are going to be utilized. They're secreted by pancreatic beta cells. Glucagon, conversely, is going to increase blood glucose levels by promoting gluconeogenesis and glycogenolysis in the liver. This is a response to low blood glucose levels and high protein levels, and it's secreted by pancreatic alpha cells. As you can see, the mechanism of action that these two hormones are going to create are exactly opposite to each other, hence why they're called antagonistic reactions. There are some other hormones we'll go into real quick. Glucocorticoids increase blood glucose in response to stress by mobilizing fat stores and inhibiting glucose uptake. This will increase the effect of glucagon and catecholamines. Catecholamines are going to promote glycogenolysis and increase the basal metabolic rate through the sympathetic nervous system. And lastly, thyroid hormone is the regulator that affects the other metabolic hormones. It has a direct impact on the basal metabolic rate. Within the thyroid, T4 is going to be converted to T3. T3 is noted as being more potent but with a shorter half-life. Let's go into some tissues and how their metabolism differs or is similar to the others. The most integral metabolic tissue has to be the liver, and it's comprised of hepatocytes. Hepatocytes maintain blood glucose levels through gluconeogenesis and glycogenolysis. They also respond to pancreatic hormones. Other functions of the liver include storing away nutrients, processing lipids, cholesterol, bile, urea, and toxins as well. The liver is obviously incredibly important, and without it, we would not function very well. Adipose tissues, or fat tissues, will store lipids under the influence of insulin. As insulin triggers their uptake, under the presence of high amounts of insulin, they're going to take in external nutrients and store it away. The release of lipids will happen under the influence of epinephrine in states of desperate need of energy, or low energy overall. Muscles are going to have very different approaches as well. Resting muscles will conserve carbohydrates in glycogen stores, and they'll use free fatty acids from the bloodstream as well. Active muscles, however, will possibly use anaerobic metabolism, oxidative phosphorylation, direct phosphorylation from creatine phosphate, of energy reserve, or fatty acid oxidation. This will depend on the fiber type and the duration of activity. If you remember, there's fast twitch and slow twitch muscles, and it just depends on whether you need a quick burst or something more long term. Cardiac muscles will use fatty acid oxidation in both the well-fed and the fasting state, so this makes it very different than the other types of muscle groups. The brain and the nervous tissue is going to use glucose in all fasting states until absolutely necessary, that it uses ketone bodies. This will happen during a prolonged fasting state, not before. Our body is generally going to try to preserve our central organs and our brain and allow the other functions in our external extremities and less valuable functions such as sexual ability to decay first before we remove resources from the central area. Obviously, the brain is the most important organ in the body, so whatever resources are available are going to go to it. We'll finish the discussion there. I know it was brief, but now let's go into some of the ways that we can analyze metabolism in a lab. There are different ways to measure metabolic states, and we'll go ahead and go into those right now. The first method is calorimetry. Calorimeters can measure the basal metabolic rate based on heat exchange with the environment. Human calorimetry makes use of large insulated chambers with specialized heat sinks to determine the energy expenditure. Respirometry is going to measure the respiration rate. We can tie how much we breathe to how much energy we're using. Consumption tracking is just simply looking at how much we eat and how much we excrete. This can also determine how much energy is being used by looking at what kind of food we're eating and what kind of energy that's going to provide us. And lastly, blood concentrations of substrates and hormones. By checking blood levels, we can determine what types of hormones and substrates are within our blood, thus giving a good indication of what state we're in. And lastly, the respiratory quotient, which is the composition of fuel actively consumed by the body. Body mass regulation is a very crucial function that our body has to go through, and there are different ways to do it. It's regulated by leptin, ghrelin, and orexin. Leptin is a hormone secreted by fat cells that decreases appetite by suppressing orexin production. Orexin is going to increase appetite, and it's also involved in alertness and sleep-wake cycle. If you notice, leptin and orexin are often considered antagonistic because of this reason. Ghrelin is secreted by the stomach in response to signals of an impending meal. Sight, sound, taste, and smell are going to act on its release. Ghrelin also increases appetite and will thus stimulate the secretion of orexin. Long-term changes in body mass will result from changes in lipid storage. So obviously, if you're overweight or even obese, you're going to have lots of lipid storage. And as you lose that, you're going to see changes in body mass as these stores are used. Changes in consumption versus levels of activity will also result in changes to body mass. If you're eating more than you use, you're obviously going to store more of it away, thus gaining weight. And the opposite is true. That's it. You've made it through the biochemistry module. Congratulations. Hopefully, this was informative for you. Study hard. Biochemistry is a very heavily tested subject on the MCAT, so make sure you have a good grasp of it. We'll see you in the other modules.